Go ahead.
Okay. So for those of you that don't know me, my name is Anson. I'm a second year chemistry PhD student and current interfaces trainee in Romy Amaro's lab. And today I'm going to be talking a little bit about how we are using multi-scale simulations, molecular dynamics and Brownian dynamics to estimate kinetic rate constants for protein-protein interactions. So to start, the question that kind of motivates our work in simulations is how we model different scales. So obviously, biological processes span many orders of magnitude, both in time and length, right? There's very short time scale and short length scale interactions between proteins and atoms, and then on a larger scale, there's interactions between cells and all the way up to whole organisms. So at each of these levels, we can, and I'm sure we're all kind of familiar with the way that we model them. We can have like atomistic representations of proteins, or we can have, larger, more coarse grain representations. Right? Models are integrated all throughout science, but simulation, the level that we simulate at, depends on the level of detail that we're modeling. So if we want to look at dynamics with simulation at an atomistic level, we have to include a description of forces to get those dynamics on an atomistic level. We have to include forces between atoms. At higher levels, like coarse grain representations, where maybe one protein is represented by one particle, now our forces are only the overall forces between proteins. We're no longer able to model lower scale interactions between atoms. So the method that we choose to model our system and simulate it to look at dynamics of the system depends on the level of complexity that we're looking at. And so today I'm going to be talking about two simulation techniques that we use. One of these is molecular dynamics, and then one of these is Brownian dynamics. And we're using both of these in our approach. So the first of these is molecular dynamics, which some of you might be familiar with. This is an atomistic representation, and these simulations, we've been able to use them for scales ranging from angstrom length all the way up to micrometer length, and femtosecond time all the way up to milliseconds. And this has been facilitated by just massive increases in computational power. That's what allows us to simulate at these levels and kind of push to higher length scales and time scales. Ultimately, what drives molecular dynamics is that this is a classical physics representation. So the forces it's just force equals mass times acceleration. We have a bunch of particles in the system, and the way that they move is based on the force acting on each of these particles. So here on the right, I have kind of one of the simplest simulations you can do, which is just a box of water. You can see each H2O molecule here, and then the motion, the dynamics is coming from the forces acting on each of these particles. So one question that might come to mind from this is, how do we calculate those forces if we're not actually modeling explicitly the electrons and the quantum level? And so what drives molecular dynamic simulations is what's called a force field. And this is a description of the potential energy, and then we get the force from just the gradient of this potential energy. And the potential energy is basically the sum of the potential energy on each particle coming from its bonds with other particles. For every pair of three that are bonded, the angle between them. And then for every four, the torsion angle. And these are what are called the bonded terms. So these are the forces between all bonded particles that contribute to the overall potential energy for each particle. And then there's additionally the non-bonded forces. So the non-bonded forces are the electrostatic interactions, so more long-range interactions between charges, and then van der Waals interactions, which are the short-range interactions, again, between non-bonded atoms. So, and the way that we get this information, the way that we get the potential energy based on bond length, based on angle, is from experimental data and also quantum calculations. So this is kind of built into the force description that allows us to do molecular dynamics simulations. The way that this is relevant to biological systems, one application is protein folding. So even though this is a cartoon representation of the protein, this actual simulation has all the atoms built in as particles, and there are water molecules as well. And the motion that you're seeing is the forces acting on all of the particles from the water, from the interactions between residues in the protein that ultimately leads to it reaching its folded state. Another very common application of this is drug binding. So this is a simulation, and here you can actually see every single atom modeled in. So inside the water box, there's a protein and a small carbohydrate, and you can see the interaction between the carbohydrate and the protein as they get closer and closer together, and eventually the protein binds to the carbohydrate. So this is a pretty good analogy for drug binding, where we have a receptor and then a small molecule drug, and we can use molecular dynamics to look at for example, the free energy of binding. We can get a lot of thermodynamic data from these simulations that matches up to experiment, and that's the main application of this, is that we can look at things that we can't get necessarily with experiment, right? We can't with a microscope observe that level of dynamics between the protein and the carbohydrate, but with molecular dynamics we can kind of get a computational microscope to see what's going on. The issue with molecular dynamics is because it's relatively detailed, because we're modeling in every single atom and the forces between every single pair of atoms, that's computationally expensive. And so we're not actually able to simulate, for example, the whole residence time of a drug molecule. So you can see here, this is what we're able to simulate in terms of on rate and off rate for drug molecules with MD. And this is what's actually clinically relevant, right? So a drug might be a very tight binder and stay bound for a very, very long time. And we just can't simulate that with MD. Despite how much better computers get, the timescales are just too long to simulate with molecular dynamics. So we need another level of maybe lower detail, but faster time to model these interactions. And one of these is Brownian dynamics. You might have heard of Brownian motion because it's not unique to proteins. They first discovered Brownian motion in like the 1800s. They were looking at pollen in water under microscopes, and you could see this kind of random walk motion of the particle in water. And what that's coming from is the collisions of the particle with the fluid. So, right, it's not just water, it models any fluid. You can model, particles in air, in gas. But ultimately what happens, regardless of the fluid, is that the motion of the fluid, sometimes when the particle collides with the particles of the fluid, either it transfers a little bit of momentum and the particle moves faster, or it slows it down and it moves a little bit slower. So that's what drives this kind of random walk. But there's also the damping force of the fluid. So the motion of the particle through the fluid is resisted by the fluid itself. And so what Brownian dynamics does is we take this microscopic level where each of these interactions with the water is actually explicitly modeled like an MD, and we use a continuum representation where we include a random force that models that kind of random motion due to those collisions, and also a damping force that is the resistance to that motion by the fluid. So what this looks like is the force acting on the particle is resisted by the diffusion coefficient. And then we also have this random force that we add to that. So the change in the position of the particle is based on the force acting on it, resisted by the fluid, and this random motion. And this can actually be applied to proteins. And the way that this looks when we apply it to proteins is we use a rigid body representation of the protein. So we no longer have all that motion of the side chains. It's just a solid object that's the shape of the protein. And for a drug molecule or another protein, it's moving through this continuum representation of the water. And when it gets close, it's bound. We can observe that. The issue is that it doesn't capture the flexibility that we get with MD. With MD, we have all of the flexibility of the protein. We can see it move. If there's like a conformational change when the drug binds, we can observe that. With Brownian dynamics, we can simulate the association of this drug molecule or another protein, but we can't see the actual conformational changes at the binding site. And that's why it's more inaccurate at close distances, but it's so much faster that we still want to be able to use this method. It's much faster because we're not modeling every explicit water molecule. We're not modeling all the flexibility. It's just less accurate. And so this is an example of what a Brownian dynamic simulation looks like for a protein and a small molecule. So what we want is we want to use a combination of these two methods. We want to be able to use molecular dynamics when the molecule that we're looking at is close to the binding site, and Brownian dynamics when it's far away, and that flexibility doesn't matter. That rigid body continuum water approximation is sufficient enough to describe its association, and then when it's close, we can use molecular dynamics. So this is the basis of the Seeker method that was developed in my lab. Again, we divide the simulation space into a molecular dynamics region and a Brownian dynamics region. And then if you're interested in the actual implementation, we use the Brown Die program, which is a Brownian dynamics simulation program for when the drug or the protein is close, is far away from the receptor. And then we use OpenMM or NAMD, which are both just MD simulation engines, when it's close to the binding site. Now, a question that might come up is how do you connect these two separate timescale simulation methods? You can't just, right, there's no way to take a trajectory from Brownian dynamics and just instantly transform it into molecular dynamics and have it continue. So we use this thing called milestoning theory. The way that milestoning theory works is if you divide the space up into what are called milestones, and then you keep track of how long it takes to go between each milestone, and then you keep track of how many times, right, the particle crosses each milestone, you can basically add these up, and you can do a little bit of matrix math, and you can determine the on and off rate from the statistics in each regime. So in Brownian dynamics, we can keep track of the trajectories, and in molecular dynamics, we can keep track of the trajectories, and we can kind of combine them using milestoning theory. So the way this works is we take our structure for our receptor and our ligand, and we define a variable that describes their binding. So for a small molecule, distance is sufficient for this. Just when it's close, it's bound. When it's far away, it's not bound. So we define this distance variable, and then we divide this region into the Brownian dynamics region and the molecular dynamics region. And then we divide that region further into these milestones that we're keeping track of the time it takes and how many times it crosses each of these. And then from these trajectories, we can kind of piece together what the on and off rate for this small molecule is. Now, the next step in this project is applying this to protein-protein interactions. And this method, I think, is pretty well suited for this because one of the main aspects of protein-protein interactions is that they kind of diffuse until they're close together, and then they form this transition complex. And then from the transition complex, shown here, there's just slight conformational changes that lead to the final bound state. So there's the diffusion till they're close together, which is what, very accurately modeled by Brownian dynamics. And then there's the close together conformational changes, which are, that's well suited for molecular dynamics. So the way that I plan to test this is kind of just a test system. It's a very small protein-protein interaction system, and it's very well studied. There's, countless studies with experimental data, mutational analysis. The experimental on and off rates are very well known. And so our goal is to set up our Seeker simulations so that we can observe the diffusional association of Barnase and bar star. And then we can also calculate the off rate, which is their dissociation. And so these are the experimental rate constants that we're trying to replicate. And ideally, we'll be able to get a fairly accurate estimation that's close to these. And then there are some other systems. I plan to try this with like the insulin dimer and a couple of other protein systems. But the next stage with this is if we can accurately compute the on and off rates for protein association and dissociation, then we can use these in even larger scale simulations. So some of you might be familiar with reaction diffusion simulations, these can be used to model all sorts of processes, because the basics of a reaction diffusion simulation is just that you have particle types, and the particles diffuse according to Brownian motion, and you can define reaction criteria. So when particles are within a certain distance, they react and form a new type of particle, or one type of particle can react and form two separate particles. And the use of this is that you can do simulations with a very high copy number. For example, if you want to look at maybe in a compartment of the cell, what happens when there's hundreds and hundreds of proteins, and over time, what happens to the overall concentration of each type of protein, you can set up a reaction diffusion simulation. But what these reaction diffusion simulations depend on is an on and off rate. When the particles are within a certain distance, they react at the rate of association, and then that one particle will dissociate into two other types of particles based on the dissociation rate constant. So this is where our seeker method comes in, because we can look at two proteins and we can determine from our seeker calculations the on and off rate, and then we can use that to set up a really, really large scale simulation where each protein is one particle. And we can observe, with a high copy number of proteins, what happens over time. So some examples of these like reaction diffusion simulation engines are MCell, Smolden, and Ready. I plan to use Ready for this, but you could take these rate constants and you could use them in any of these programs. So just to kind of show what this looks like is we take our particle types. If it was Barnase bar star, like I was talking about earlier, we determine the on and off rate from seeker. And then in the larger scale reaction diffusion simulation, we say, okay, when these two particles, each representing one of the proteins, are within a certain distance of each other, they'll react according to this rate, lambda, to form particle type C, which is the bound complex. And then at the dissociation rate, they will dissociate, and that one particle will split into Barnase and bar star separately. And then we can populate the system with a high number of these, and over time we can see what happens to our system. Yeah, so this is kind of the future direction I'm taking this. The challenges, of course, are our description of the binding of the two proteins in seeker. Actually, let me show this slide one more time. So with the protein ligand description, distance is sufficient because the ligand is very small, and so the rotation doesn't matter as much as the distance does. But with the protein, there's that conformational change. So we can't just say that when they're close together, they're bound, because they might be close together and just be in that, that transient complex. So we need to use another variable or set of variables for this. This is called the collective variable. And so one way we could go about this is we can use both the distance and like a description of the RMSD from the bound state. And then we divide the milestones not just in distance, but also in conformation. And so that's one way we're going to possibly go about this. Another is to use just a set of points on the binding surface of each of these proteins. And then when they're, then that captures both distance and also this kind of rotational conformation. So yeah, that's where we're going with seeker. And yeah, ultimately our goal is to have a framework where we take a protein system of interest that we maybe aren't able to get rates from experimentally, and we can run our seeker calculations and then build even larger scale models of reaction diffusion. Yeah, so I'd just like to thank my lab, the Amaro lab, of course, my PI, Romy Amaro, And then my mentor, Lane Vatapka, he's a staff scientist in the lab, and he developed Seeker and works with me on this. So yeah, if there are any questions, I think I have some time.
So I actually don't have any questions, but that was just like that's very cool. Just out of as a compliment.
Thank you. Yeah.
Anybody else? Jillian, did you say something?
Yeah, I was wondering, are you doing more methods development type of stuff, or do you have a specific project that you're working on? I know Romy does some of the COVID vaccine stuff, or I guess COVID project and looking at that finding. Are you working on any one of those projects specifically?
So I'm not working on any of the COVID stuff right now. Obviously, yeah, that's something our lab is kind of known for, is the like the COVID aerosol simulations. The applications of this project are hopefully going to be like useful for that. If we can get this to work for protein-protein interactions, then there's all kinds of like large scale stuff we can look at. We can look at the COVID spike protein. So yeah, ideally that's something we'll get to, but right now, yeah, this project is very much just in the early stages because we're trying to adapt a method that is mostly built just for protein small molecule to actual much larger scale protein-protein interactions. But yeah, hopefully eventually we'll be able to apply it to like COVID stuff.
Cool, it was a cool talk. Thank you.
Okay, if there's no other questions.
All right, here, yeah, I can stop